Ladies and gentlemen, we will now begin. Please welcome Christopher Bowlus.
Good afternoon. Good afternoon, everyone. Welcome back from lunch. We've got the start of our general session 3 with a lot of really exciting speakers. Our first speaker was just our recent moderator. As you could tell, has a wonderful sense of humor. Dr. Heather Francis is a professor of medicine and scientific director of the Indiana Center for Liver Research at Indiana University, where she focuses on the role of cholangiocytes and, interestingly, mast cells, specific cells within the liver, which has generated a lot of really interesting data that could offer some new therapeutics. She's also the co-chair of the Scientific and Medical Advisory Committee for the PSC Partners Seeking a Cure group, and is here to talk about the impact of the PSC Partners grants and the patient organization. So, Heather.
Thank you. Thanks so much. Okay, good, it's going, because I can just see myself over here and over here. That's fine. I just want to give a little brief overview of what the money that I have received from PSC Partners has done for me throughout my scientific life. A little introduction for those of you that don't know me. I grew up in West Texas. Both of my parents were educators, so I was never given an option about what I would do with my life. That's fine. So I quickly received my bachelor's degree. Then 20 years swiftly went by. I don't know how 20 years went by. I did not really age very much, but what I realized was that I could become a very glorified lab technician or I could do something else with my life. And so I found myself back in graduate school, and in 2010, I earned my PhD from Texas A&M College of Medicine, all of this with the support of my mentor and now colleague, Dr. Franco Alpini. My dad always reminded us in class, I had both of my parents as teachers, that if you aren't gonna do anything with yourself, you will for sure end up in a van down by the river, okay? Why do PSC research? So I don't think I really need this question here. There are other avenues, there are other things to study: fatty liver disease, hepatitis, liver cancer, PBC, and I touch on some of these in some of the work that I do, but my real passion, my heart is in PSC. And I'm not gonna go through all these stats because Joanne also told me that I only have 8 minutes. So you guys know all of these facts. You know there's no cure, that you know that we science, medicine, volunteers, everyone are waiting, working to try to find better options for patients, even to manage symptoms. And we've heard a lot about that over the last few days. So my friends have no clue, right? And they think that I'm just having fun and going around and traveling and doing whatever. My mom, my dad both think that I'm just way greater than I probably am. Society thinks I'm also contributing. I don't know. My boss probably does think that I'm on Facebook, although really Instagram is my zone. And what I think I do, I think that she looks a little bit like Indiana Jones, and I like to think like that. What I actually do, though, is seriously sit and stare at computers, but more importantly, ask, beg, plead, and try to get money to fund my work and my research and keep my lab going. So what specifically, how did this happen? And this is just talking about my PSC work here. So in 2012, I submitted my first PSC Partners application, and this was from the encouragement of Dr. Green, who's also going to be speaking in a few minutes. And surprisingly, in 2013, I got funded. It was my first external grant that didn't come from a federal agency. I had a VA Career Development Award, but this was my first time to kind of be out there in the public by myself, and so that was very exciting. Then during the next few years, I generated a ton of data and started submitting an R01. So this was between 2014 and 2015, and a first time triage, second time triage, third time triage, start all over, scrap it, sent it again, and I finally got funded. And for me, that was a huge accomplishment. It is for any researcher to get your first R01 is always sort of that baby that you hold on to. Although my first child she's a little bit annoying. It's fine. So I that's my PSC work. So then in 2018, I submitted another one. That one didn't get funded. In 2020, I renewed this PSC work that I started back in 2012, in 2021, I renewed that. And then in 2022, we submitted another PSC Partners grant, and we were awarded that by the PSC Partners of Canada. And we're very excited to be doing that work using a very novel small molecule. So what are some of the key outcomes from my work? I was able to identify and really grow this concept that mast cells play a really key role in driving PSC progression. We find them in large numbers in patients with PSC within their livers. We find increased histamine serum content in patients with PSC when we compare them to control patients. We've used over-the-counter drugs like cromolyn sodium and antihistamines in our models in the lab to ameliorate damage, and these have maybe some promising outcomes potentially for a repurposing of old drugs. We do use different animal models. I talked about this a few days ago to study mechanisms of action, and we've learned a lot about how mast cells act in animal models and also how they act with other cell types in your liver, including your bile duct cells, your hepatocytes, and other immune and inflammatory cells. We've identified what we think are potential biomarkers. So histamine could be a potential checkpoint, and kinase levels are also high in patients with PSC. We've also developed other techniques to study the cell-cell interactions, including organoids, which we can also build from human liver cells. And these are things that I've only been able to do that have been springboarded by the money that I was given by PSC Partners over 10 years ago. Some of the impact here, including the scientific impact, my career, personal connections, and opportunities. So I tried to count this. There are, I think, about 15 publications generated from that first PSC grant. We presented at AASLD multiple times. We were given a travel award to my graduate student at the time, Vic Meadows. I was asked to join as a SMAC member and become a grant reviewer, and I think I actually volunteered for this, and I was jet-lagged in Europe, and I ran into Ricky in the bathroom, and I think I said I would love to review grants, not knowing that then I would also, Rich Green would also dump his job on me later, but that's fine. I'm very happy to be doing that. Lots of new collaborations. I'm building relationships with other people in the scientific community that I may or may not have ever met. I've been invited to speak at numerous places, including being invited here, which is such an honor to be amongst all of you. The community, I go to a lot of meetings. I mean, we've been on hiatus a little bit, but what I really love about this meeting is the personal connection that you get to make with actual patients that are living with the disease that you're studying. I do feel like there's a very collaborative effort here amongst the scientists and the clinicians and industry to try to work towards the common goal of trying to find something to either help with symptoms or even identify a potential cure. The dinner last night for me was super informative and fun. I learned a lot about the people at my table. They do not follow the rules. If you see any people from table 19, they do not follow the rules, okay? I don't know where Margaret is, but I'm guessing she's somewhere with her bag of Lay's in the very back of the room. But we don't usually get to do that. We don't usually get to interact face-to-face with people. So for me, this community and the impact of being part of PSC Partners, this is really, for me, like, people were saying this earlier, the bread and the butter. I love it. This is just has made all of this worthwhile. What does the future look like? I think it's pretty bright. We need samples from different stages of disease. This is a very limiting factor in the research that we're doing. I think AI, artificial intelligence, coupled with deep learning machines, these are going to be able to maybe push out some feasible targets. Again, the organoids, we're hoping that this is going to be a method we can use that's a little bit more cost-effective and be able to study the signaling mechanisms, personalized medicine, potentially from these organoids, and of course, increasing our collaborative efforts by involving those that work with the big data sets to tease out targets and potential therapies. These are the rules. I try to apply it to my scientific life, but I think we should try to apply these to everything that you're doing. Even when the door is closed, especially in science, you've got to keep knocking, you've got to keep trying. It really helps when you have a strong group of people behind you, mentors, colleagues, friends that push you, encourage you. And a group like PSC Partners does just that. Don't stop learning. Don't ever assume anything, question everything, teach others what you know, always be ready to share what you're doing. Analyze, be humble if you can. A little hard for some of the people at my table last night, but we're working on that. Take some criticism, guys, okay? Take your own advice here. Give the credit where it's due, and the credit here obviously is due to all of these wonderful people that organized this meeting, the volunteers, the staff. I can't name them all, so I'm not even gonna try. Take initiative, ask those questions that you wanna ask, and love what you do or just get out. I mean, there's just no reason to be doing it if you don't love doing it. And that's it. Thank you all so much for listening.
You guys don't see the timer here, but she hit it to the second. So that's, wow. And I don't know if it comes across, but getting an R01 that Heather was talking about is not easy. These are NIH grants, which only about 1 in 10, 1 in 8 maybe get funded. So to get an R01 and then to get it renewed, that's a big accomplishment. And as Heather was mentioning, it's made possible by a seed grant from PSC Partners. So this is kind of the return on investment you get from these grants that the PSC Partners is funding, as it leads to greater funding from federal government and really helps move the field forward. Our next speaker is Frank Anania, who is currently the, so pull up your bio again, acting deputy director, is that right? One of those things at the FDA and all the different little agencies that go down to where he's at. But Frank, before going to the FDA, was had a background in research of an expert in fatty liver disease and liver fibrosis in particular. And I have to say, it's been a huge pleasure and really, I think, a highlight for myself and I think many others here is to have Frank here to really help us understand the FDA's perspective and how we can move the development of therapeutics for PSC forward. And I'm really appreciative of him taking the time to actually give us a presentation earlier this week as well as this week, this presentation, knowing that we're not the only disease group that's asking for some of his time, and he's come here to spend several days with us. So I really appreciate Frank's participation here.
Thanks, Chris. I was thinking that I should say greetings from Washington, D.C., but then I thought maybe people throw tomatoes at me. Since living inside the Capitol Beltway has its interesting and unique aspects, I often tell people that there are more lawyers in Washington, D.C. than there are fire hydrants for my dogs to urinate on. And that actually, when we write documents from the FDA. I was a scientist, as Chris pointed out, before I joined it, but now I've learned to write like a lawyer, which is kind of interesting. So as typical bureaucrat, I have to disclose nothing to you because I don't take anything. Despite what you may hear about certain Supreme Court justices, we can't take anything and we report things constantly to the government. The views that I'm going to talk about today are mine and not necessarily those of the government or HHS or the Food and Drug Administration. Okay. So I think you've heard a lot about this, and I'm not going to spend too much time about it, but I do want to point out to you a couple. Why did I come here? There are two diseases, in my opinion, as a hepatologist, that have clear unmet medical needs. One of those is alcohol-associated liver disease, which, by the way, is now the leading reason that people undergo liver transplant in the United States. But the second one is primary sclerosing cholangitis. And because, as Dr. Francis pointed out, there's so few people, 30,000, maybe 40,000 Americans afflicted with this terrible disease, it is considered a rare disease. And I will just point out to you that my division, the Division of Hepatology and Nutrition, DHN, Its portfolio does not include cholangiocarcinoma, hepatocellular carcinoma, or any of the viral hepatitis, A, B, C, D, E, and G, or whatever others that they've come up with. We mostly, except for NASH, have rare diseases as outlined here in this, let's see, in the list below. I don't know that I have a good pointer for you, but anyway, I'll figure that out in a minute. So how does the government define a rare disease? The government defines a rare disease by a disease that afflicts less than 200,000 Americans, and that's the Orphan Drug Act was passed in 1983 to help induce industry, stimulate their interest in many rare diseases. So in our division, most of the diseases are rare, including PSC. Okay, so one of the reasons why this was done, of course, was to provide industry financial incentives. And one of those includes a 7-year period of market exclusivity. So that means that no one can develop not just a generic drug, but what we call a 505(b)(1) drug. They can't do that. It provides tax credits, and the law enables the FDA to designate these drugs with orphan status, and that has another financial or monetary award to it, if you will. I just want to point out on the next slide how we've done with this. So, because I know that many of you are probably very frustrated with the glacial pace in which we have not yet approved any drugs. But I do want to point out that the Orphan Drug Act has been rather useful. And I just pulled these data from the Office of Orphan Drugs. And as you can see, in the 4 decades since the Orphan Drug Act was passed, the agency has designated over 6,000 drugs as orphan, they've studied over 1,000 rare diseases, and we've had 882 approvals as of July this year for over 350 of those diseases. So the agency now considers that about 5% of all rare diseases in the United States, as classified by the definition we gave you, have at least one marketing approval. My hope is before that I retire from the agency that we do have a drug for you. Now, back in the height of the pandemic, I believe that PSC Partners had a patient-focused drug development session with us, or PFDD. There are a lot of acronyms in the FDA. You have no idea, but that's one of them, PFDD. And I think you all met with us in October of 2020. And one of the things I want to point out, I'm going to show you concrete examples, not just talk about the bureaucracy, of how we actually use patient-focused drug development tools to help us approve some recent drugs for other rare diseases. And as you know, we engage directly with patients, and when a particular drug comes to us for consideration in its development, not necessarily to be marketed, what kind of issues that the industry brings to us and patients bring to us, and we can work together. And I want to show you how we've done that recently. Okay. There's also a number of other programs that have come out. This is the Rare Disease Endpoint Advancement Pilot Program, which is very new, RETA. This was an outpost of a law that was enacted last December, the Food Drug Administration Reconciliation Omnibus and Reform Act. It has another abbreviation called FDORA. And one of the things that came out of that was that this rare disease endpoint advancement is piloted, and that's very important to scientists, clinical scientists, and the drug manufacturers, because the statutory requirements to get a drug approved for most indications are rather rigorous. You have to have 2 large-scale, randomized, double-blind, placebo-controlled trial with thousands of subjects. And so that obviously does not apply and cannot apply to a rare disease. Also, the duration of those trials are very long. But here, we know that we cannot take trials on for a long time. So this is and I just want to point out to you that any slide that I present in the public to you are available to you. So the organizers can give you these slides, and the hyperlinks are not dead, they're alive. I make sure that they're always alive so people can see them and go on to the website of the FDA, and I would encourage you to do that. Okay, so there are a variety of documents. The government is very good about producing guidance documents, trust me. We do this all the time, but they actually have a lot of utility. They give people ideas about what we're doing. It helps industry, helps academic investigators who want to do clinical trials and so forth to understand exactly what we're doing. So we do think the patient voice in rare diseases is very important. I want to emphasize that point. Okay, now, how do we use these concretely? And the one major way in which we do this is using patient-reported outcome, or hereafter PROs. So patient-reported outcomes capture how you feel, right? Because the idea in the FDA is that you want a therapy that improves life expectancy, that makes you feel better, that returns you to work if that's what you want to do. So the idea always in our brain is etched in there from the day you get there. It feels, functions, and survives. That's what you want to do. You want to see that a drug does that, whatever the condition. Okay. And so these patient-reported outcomes can be developed as well as observer-reported outcomes. So unlike a patient-reported outcome or an observer-reported outcome is done by a third party. And oftentimes in rare diseases, we can use these reported outcomes from patients or observers to help us define an endpoint. And in a clinical trial, as you all probably know better than I, it is the endpoint, to meet the endpoint in a statistically and clinically meaningful way, that you get efficacy. Okay. And so the bottom line, as I just mentioned to you, is it's all about clinical benefit. We look at drugs basically and do a benefit risk assessment. So if there are a lot of safety signals with a drug, doesn't really matter how good the drug is. If the drug hurts people or causes drug-induced liver injury or causes, an arrhythmia in your heart, we're not likely to approve it because the benefit risk, usually we don't. And it's because the benefit risk may not weigh toward the benefit. So that's really very important. And so, in liver diseases, there are clearly outcomes about these as irreversible mortality and morbidity that may develop over many years. And we understand in the division that liver diseases, particularly this one, wax and wane. They ebb and flow. They have exacerbations and remissions, much like lupus does in the rheumatology field. So PROs can be selected as a primary endpoint or a co-primary endpoint, and there's different ways to do this. And so we have a whole development pathway or strategy, how we partner with industry and with patients in rare diseases. And I'm not going to spend a lot of time going through all these details. Again, some of this is just down in the weeds bureaucracy, but I just wanted you to understand that there is a mechanism in place. Now, I don't want you to get upset about the molecules. Heather Francis did a spectacular job as a basic scientist. She didn't put any cells or molecules up, and I now feel badly that here I am a bureaucrat and I brought these cells to you, sadly. But that's not the reason why I did it. And you don't need to read anything on here, except for the fact that PFIC is another cholestatic disease that's rare. It affects primarily children, and they itch like mad. And in fact, they have other issues too, but the itching is probably the worst symptom, okay? And there are genes and causes, there are all kinds of mechanisms that can trigger this. There are different subtypes, not important to you, but the itching, remember the itching. Okay, and another syndrome, I don't have any cells for this one, but the Alagille syndrome is another very rare disease that primarily affects children. And again, like you, patients have cholestasis. They can also have other organ involvement, including the heart, skeletal muscle, their eyes. And the treatments, there's no treatment for the itching, but again, the itching is very, very common. And it can present very early on in life, even in the newborn. And it can be quite burdensome for patients. Oftentimes, babies do not get adequate nutrition because they're so uncomfortable. So these were two diseases that are cholestatic, liver diseases, rare diseases, no treatment for either one of them. And it turns out that we have a therapy now, and and in order to figure out how to measure its effectiveness, that's where we turned to the patients, because this was an unmet medical need. And so what we did was we did meet with patients and their families, since these were children. And we felt, and as I tell you, patients are often the best experts in helping us develop and industry develop patient-reported outcomes. And so one common thread across these various groups of patients with cholestatic liver disease in the childhood was this itching and how it impacted their lives. And so we, with the industry, developed observer-reported outcomes. So, and what we mean by that is we had a third party, namely the caregiver, with electronic diaries, come up with a scale, which the industry and the FDA outcomes assessment team collaborated on. I'm not an expert at that. And they developed a measuring device, a tool, an instrument, a reported outcome for parents and caregivers to see how patients would or would not improve on IBAT inhibitors. And if you don't know what those are, they're ileal bile acid transport inhibitors. They block the uptake of bile salts in the small intestine. And we basically, how did we know whether they worked? We did some analysis. And what we found was that what patients reported at 8 to 12 years old was identical, highly concordant to what the parents were reporting. So I wanted to go back to that. So as you can see, we just didn't assume that they would work. That is the observer-reported outcome. But we wanted to see whether or not those reported outcomes were identical to what an older child or an adolescent reported. In fact, that's what we found. So this was some of the studies that we did when the data came in. Now, I will just point out to you in the last minute or so, both of these diseases, the itching was the two drugs were approved, odevixibat and mirabexabat, in the summer of 2021 for the indication of pruritus in these diseases, respectively. The point is that both of the studies that were done were very small, randomized, placebo-controlled trials. In fact, one of them only lasted like 12 weeks. And what I want to leave you today is that it is people like Dr. Francis and Dr. Green, in my opinion, who hold the keys for industry's development of drugs for you. Because most of what the people in the country often forget is that the basic science drives drug development. I really mean that. There are some people here from NIH today, I think. We would not have had COVID-19 vaccines if it wasn't for the NIH. We'd still be walking around with those masks on. But it was because of their interest in respiratory syncytial virus and HIV through the Ryan White Act that they had been working on these RNA vaccines for almost 20 years. So when people ask, how did these things get developed so fast? The science was known. And I tell you that in this disease, it is these bright people who are doing the work of science that will make breakthroughs that ultimately industry will carry forward in conducting trials. And hopefully, I think we're in a very exciting time in hepatology. I'm very optimistic about this disease and others. And I'm happy to be here and thank you for your attention.
Thank you, Frank. That was excellent. Our next speaker is Dr. Richard Green, who I think you all remember from last night, getting a nice award. And he is a professor of medicine at the Northwestern University Feinberg School of Medicine, and both a clinician with an interest in PSC as well as a basic researcher in the molecular mechanisms of liver disease. And he has been the co-chair of the Scientific Medical Advisory Committee for PSC Partners for, we decide, over a decade. Oh, she's just got done. And I think your talk is to talk about that experience?
Yes. Okay, thank you. Ricky, I want to thank you, and I want to thank all of you for coming, because it's the reason I am doing what I'm doing is because you're out there. And this is a true story that happened on Tuesday when I went to visit my mother in Scottsdale, Arizona. My mother doesn't consider me a doctor. I went to med school, I didn't become a surgeon and go and practice with Richie Gordon. I have no idea who Richie Gordon is, I never did, but because of that, she asked me the question she's been asking all along. She's like, Richard, are you still doing that bile thing in your lab? And I'm like, yes, mother. She's like, why don't you do colonoscopies in the western suburb? I don't want to, mother. You make more money. I know, mother. And I'm sure when I talk to her tomorrow to say I landed, we'll have that same discussion. But there's a reason I don't want to do colonoscopies all day in the western suburb. Because like almost everybody's talking to you, I'm passionate about what I do. I couldn't get up in the morning on Monday and say I've got 35 screening colonoscopies. I could, but I don't want to. So what I do is I take care of patients with cholestatic liver disease and fatty liver disease, and in my lab, I look for insights to how we can treat them better. So PSC Partners has had a research grant program, and I counted it. There's been over 75 recipients. And this is from their website. You can see patient education and support is important, patient registry is important, and then research grants. And that's pretty much what I'm going to talk about and how the research grants fit in the big scope of things. So just briefly, PSC Partners and affiliated PSC Partners Canada offer 2-year seed grants to conduct important and novel research. This could be basic, translational, or clinical research, and I'll explain that. They have to be related to PSC or closely related diseases such as IBD or cholangiocarcinoma. The awards are for established investigator, but one grant is available for young investigators to help develop the next generation of researchers. Now, in 2022, PSC Partners awarded 3 international studies, plus 1 additional grant was awarded by PSC Partners Canada, and they've awarded over $6 million since the program began, which is really outstanding in developing the field. So it's an international program. You submit your grant, and then it's evaluated in same as we would if we submit a grant to NIH by what's called the Scientific Medical Advisory Committee. We dole out the grants to about 15 researchers. Some are clinical, some are computational, some are research, and 3 people review it, and we talk about the good grants, and we rank them scientifically based on the quality of the science and whether we think it's important. And then based on these recommendations, the board of directors will review all applications and award the most promising studies. It's usually, but not always, the most scientifically best one because there are priorities. And if it's a good grant and it's related to cardiology, it's not going to be awarded. So this encourages investigators to conduct research in promising new areas by providing seed money, venture capital, with the goal of the study results will lead to federal or other funding. Much of the federal funding here in this country is through NIH or even the VA, but there's funding agencies throughout Europe, Britain, I guess they're not in the EU, and Canada. So what is seed money? Seed money is for future investigations. In order to have a grant awarded by NIH, for instance, it needs to be really significant, it needs to be innovative, but it needs to be feasible. And if something has never been done before, it's very innovative, it's very significant, you're gonna get a criticism saying, great idea, but I'm not sure it could be done. So what seed money does, it allows you to do the initial proof of concept research and get the preliminary data to make you highly competitive for much bigger grants. Research is expensive. It's in millions of dollars. So NIH grants or VA grants or grants in Canada and Europe, they're multi-year, and you've got to have a good grant, and there's got to be a reason for people to accept it. And they particularly like high-risk, high-reward projects. And this provides a concept, not a proof of concept. And again, this is like venture capital for academia. It gives you the money to start the process and see if you get buy-in from a bigger resource. So I'm going to talk about clinical translation and basic research. There are 3 distinct but interconnected and complementary domains in the field of medicine and biochemical research. And the distinction is actually artificial, it's oversimplified, they overlap, but the time to clinical advances may vary with these type of investigations. And that's very important when you're choosing the priority for funding a grant. Now, you've heard all day about clinical research, which is in blue. This is one drug versus another. It's the way we test medicines before they come on the market. We want to make sure they're safe and effective. And this is virtually always the final step to bring it to clinic. But over the past 25 years, although we know that PSC's an immunologic disease, it's related to toxic bile acids, it's scarring. When we've used immunosuppression and URSO and antifibrinolytics, they don't work. Well, why do they don't work? Because they don't work. And I'll get back to that later. I don't know why they don't work. They should work, but they don't. So this brings up what Frank was talking, and actually Heather, basic research. So basic research is basically fundamental biology relevant to the liver or the immune system or PSC. I was looking for a quote and I couldn't find one, but actually I went on ChatGPT a couple days ago, and it says, basic research is the compass that guides the voyage of clinical medicine. Without understanding the intricacies of biology at its most fundamental level, we navigate the complexities of healthcare blindly, missing the opportunities for true innovation and effective treatment. ChatGPT, September 5th. As my daughter would say, what a wonderful robot quote. So basic science research focus on the understanding of the principles of biology, physiology, genetics, and life sciences. You want to find the fundamental molecular and cellular level, organisms level. It's the foundation for further biomedical knowledge, and it provides insights that can lead to breakthroughs in understanding disease processes, disease pathogenesis, developing rational new treatments, and advancing medical science. Translational research is bench to bedside. We take it from the lab, we put it in the clinic. And it bridges the gap between scientific discoveries and basic research and clinical settings and real-world scenarios. It plays a critical role in getting novel ideas, innovation, and advancement. And basically, it translates the findings and insights from fundamental research with tangible benefits for patients, communities, and society. These are treatments, therapies, drugs, genetic treatments, diagnostic tools, and medical therapies. I think you all know what clinical research is, but there's a range. It could be a controlled trial of 2 therapies. It could be a diagnostic methodology. It could be a prevention strategy or epidemiology. It could be quality assurance, or it could be to improve diversity, equity, and inclusion. In other words, this example is treatment A better than treatment B? So I want to point out that, unfortunately, since I started in this field 30 years ago, we do not have effective therapies, and you all know this. This is PSC partners seeking a cure. And I think everybody here, you keep asking what will happen in 5 or 10 years. If I was going to ask anybody who doesn't want a cure in 5 to 10 years to stand up, no one would. And to be honest, I don't think there's going to be a cure in 5 to 10 years. And that's not my goal of research. What I want to do is find some medications that are safe and effective that you can give to people with PSC, and their disease will not progress, and they won't need liver transplants. So I think much sooner than a cure, we're going to get effective therapies. And I'm very optimistic about this in the next 5 to 10 years, and hopefully sooner. So maybe it shouldn't be PSC seeking a cure. It should be PSC seeking a highly effective medicine to treat patients who don't need liver transplants, but there's no acronym for that. So if we can find a medicine like to control cholesterol or to control hypertension, people live normal lives. So I think finding a treatment is a shorter order way than finding a cure. A cure would be great. I want a cure, but I'm optimistic about treatments. So I'm going to just very briefly go through some recent PSC Partners grants to give an idea of what it is. Well, one awarded this is Unbiased Tissue Metabolic Profiling of PSC IBD. It's looking at metabolism in these diseases. PSC studies on a bile duct on a chip. They're developing chips like little, 2 by 2 centimeter or 2 by 2 millimeter chips that can help us do experiments of bile ducts. The next one is on PSC liver wait list and post-op outcomes. This is a test, this is probably a study, a short-term study that can be completed while the first 2 studies may take years. And the last one, or two other ones are hepatic unfolded protein response and murine bottles of PSC and novel proteome-wide antibody discovery in PSC. These are basic science. They're all also Young Investigator Awards. So what research should be funded by PSC Partners? This is my opinion. I don't know. It's not my call anyway. But I have some suggestions for an approach. And it's up to the people at PSC Partners to figure out whether they want to listen to the suggestions or not, or do whatever they want. But first of all, the fundamental requirement is the science must be sound. If we're going to fund a grant, we want to fund a grant that will answer the hypothesis or question. It may be yes, it may be no, but the science must be found. And then there's essentially value decisions should be made. Do we want a high risk, high reward grant that may take several years? Or do we want to do multiple incremental advances? Do you want to hit a grand slam and you may strike out? Or for Europeans, do you want to get a penalty kick and win the World Cup or have an own goal? But the ones you will fund will have different implications on when they're done. Single PI, multi-PI, multi-institution. Generally now science is not done with a single PI. It's too complex and we always have other collaborators. What are your short, long, and intermediate goals? And I said before, basic, translational, or clinical science. And then you want to know, do you want a finite project that can be completed in the 2-year grant period, or do you want to give something seed money for larger awards? And lastly, do you want to develop the next generation of investigators who may then be focused on PSC? So I don't know the answers to all of these. I know the answer that science must be sound, but these are all important questions and will have tremendous implications in the time course, et cetera. But again, I am very optimistic that we won't have a cure, but that we'll have a highly effective medication that may be non-toxic and prevent any of you from progressing and needing liver transplants. So I'll stop right there.
That was excellent. Thank you, Rich. Our next speaker is Lisa Forman. Dr. Forman is a professor of medicine in the Division of Gastroenterology and Hepatology at the University of Colorado, where her main clinical interest is in autoimmune liver diseases, including PSC. She's been well published and has been active in a lot of the societies, and including being a member of our writing group for the ASLD practice guidance. So, Lisa.
Thank you. So if you are upset about Vanco, you can blame me. So I don't know how I got lumped with the research section, but I'm going to be talking primarily clinically about the state of liver transplant and hepatitis C. Whoa, PSC. No disclosures. So I think once someone is diagnosed with PSC, all these questions run through their head. Am I going to need a transplant? How will I know? What will happen afterwards? And although I think it's impossible to predict the trajectory of any one patient, I think in general, people do well for PSC for a while, and then all of a sudden hit a wall, and then it becomes obvious that they need a transplant. Overall, I'd say 50% of patients will end up needing a transplant, but on the other hand, 50% of patients won't. Initial studies actually showed that transplant-free survival for PSC was actually quite low, but if you look at more population studies that are from non-academic centers, the survival without transplant is actually much better. These are general indications for transplant. If one has a MELD score of 12 or above, if a person has any complication of cirrhosis regardless of MELD score, patients with cholangiocarcinoma, which we'll talk about, and then often with patients with PSC often are sicker than their MELD score. So quality of life being affected, recurrent cholangitis, pruritus, that's also an indication for transplant. I want to just give a shout out to Ricky, and if you look at her webpage, there's actually a really great amount of information if you're thinking about transplant, what kind of questions you should ask your hepatologist. So it's important to know that really transplantation has never been more successful. To date, there have been more than 1 million organ transplants performed in the US, and there have been approximately 270,000 liver transplants performed here. And in 2002, in the United States, there were 9,528 liver transplants performed, which is the most ever. And the survival is quite great, in my opinion, with 93% adult 1-year survival and 88% adult 3-year survival. The evaluation process is quite comprehensive. Once a person has financial approval for transplant, they meet with our team and they'll meet with hepatologists, the surgeons, they'll have extensive labs, they'll have an evaluation of their heart, they'll have a general health assessment, anesthesia may see them. Some people will meet with our transplant psychologists, everyone meets with social worker, they meet with nutritionists and infectious disease. And then if all of us get together and we all basically discuss all the parts of the evaluation, and then as a group, we decide whether or not we think you're an acceptable candidate to list for liver transplant. Well, once someone is listed for transplant, how are organs allocated? And the way that we allocate organs is we have a policy of sickest is higher on the list. And the way we define sick is this MELD score, which basically predicts 3-month mortality. It ranges from 6 to 40. Whoever has a higher score is sicker and therefore would be higher on the transplant list. So for example, if you have a MELD score of 26, that puts you at a 20% mortality rate for 3 months and would be much higher on the list, for example, than someone has a MELD score of 10. Now, the MELD score has gone through many permutations. Most recently, we incorporated sodium, so it's the MELD sodium. And then as of July 15th, actually, before I tell you the news about July 15th, we can tell you that since the MELD system was put into place, the 90-day waitlist mortality is about 30% lower. And luckily, there's been no change in post-transplant outcome. So here's what happened as of July 15th, is there's a new MELD score, and it's called the MELD 3.0. And basically, we realized that there are some people that were being disadvantaged by the current MELD system, in particular females who have less muscle mass and, for example, will have lower production of creatinine, and therefore their MELD scores tend to be lower. And so the MELD score was revamped, some of the coefficients and the different variables were changed, interaction terms were added, and again, now we have MELD 3.0, and in general, it now gives the females 1.33 additional MELD points. If you're interested in calculating your MELD 3.0 score, there are lots of websites that you can go to. This is one that we use, and it not only calculates the current MELD 3.0, but if you want, it also shows you your previous MELD and the MELD sodium. So what about MELD exceptions? So there's 2 situations for this, for you guys, that this will come into play. The first one, you might think that as a patient with PSC, maybe you're disadvantaged because you primarily are having episodes of cholangitis, but you're not really able to develop problems with your clotting factors because your liver is actually functioning well. So we did come up with some exception criteria, and the patient with PSC to have this upgrade has to have cirrhosis, And they have to be admitted to the ICU at least 2 times over the period of 3 months. In addition to those 2 criteria, they have to have a stricture that's really not responsive to PTC, for example, or ERCP. And in addition, the patient has been diagnosed with a resistant organism. There's also now exception rules for patient with bile duct cancers. And in the very beginning of liver transplant, we didn't even think about transplanting people with cholangiocarcinoma. And thanks to the work done by Mayo, we've really figured out that there is a subset of patients with cholangiocarcinoma that actually can do very, very well with transplant. And so if a person has an unresectable hilar cholangiocarcinoma, and the diagnosis can be made either by a biopsy showing that there is cancer, or the CA19-9 is greater than 100, or on FISH analysis, it shows aneuploidy. If there is a mass lesion, it must be less than 3 centimeters. There should be no evidence of any extrahepatic spread. These people can be candidates for this exception to get a MELD upgrade, but they have to undergo treatment with radiation and chemotherapy as outlined on the slide, which is known as the Mayo Protocol. So if someone meets the PSC criteria, or if they meet the cholangio criteria, you take the median MELD at the institution minus 3 points, and that becomes your MELD score. So if your lab MELD is only 7, but the median MELD is 30, once you go through the protocol, for example, your MELD automatically would go up to a 27. How do people do after liver transplant? Well, in general, patients with PSC do really, really well. As you can see from this figure here, PSC is the solid line above, and so patient survival tends to be quite good compared to other indications for liver transplant. This was a study that recently came up, and it is from Europe, but the data is similar to the data in the United States, and you see that there's excellent both patient, which is in the green, and graft survival, which is in the purple. So 1-year patient survival of approximately 89%, 5-year survival 80%. What about recurrent PSC? I know this has been a topic of conversation both yesterday and today. And it occurs in approximately 20 to 30% of patients that we know of. And how do we diagnose recurrent PSC? Well, a person has to be at least 90 days out from liver transplant. They have to have an MRCP or ERCP with the typical beating and strictures. If you were to do a liver biopsy, you see the typical onion skinning representing the fibrosis of the bile ducts. And as mentioned earlier, you have to exclude chronic rejection and vascular conditions such as hepatic artery thrombosis. Now, I think it's still very unclear what the risk factors are for developing recurrent PSC from those that don't. We think people that have an intact colon, people that have active inflammatory bowel disease are at higher risk for developing recurrent PSC. Also, history of rejection seems to maybe play a part of it. Males more than females, a history of cholangiocarcinoma as well. Now, despite PSC occurring again after transplant in approximately 20%, it's important to note that first line that most patients do really well. Excuse me. And the treatment of recurrent PSC is very similar to the treatment of regular PSC, giving antibiotics if and when you develop an infection, stenting as needed. But up to 25% of those with recurrent PSC after liver transplant will go on to need another liver transplant. And as we'll talk about, there are generally better outcomes for repeat liver transplant for PSC compared to other indications. So this is, again, an international study looking at the survival rate after liver transplant with relative to when a person develops recurrent PSC. So if you develop recurrent PSC early after liver transplant, you tend to have a worse prognosis. So if you look at the figure that's in purple, that's looking at survival if you develop recurrent PSC within 90 days, and then the green is recurrent PSC after 10 years, and you can see that those people tend to do better. Looking at, in general, retransplantation for recurrent PSC, both in graft and patient survival. If you look at other indications for retransplant in patients with PSC, when recurrent PSC is the indication, the survival tends to be better. And then if you look at outcomes of recurrent PSC retransplant compared to primary liver transplant, once you get to about 5 years, the outcomes are very similar. What about outcomes for patients that were transplanted for cholangiocarcinoma? Well, this is all data if you fit into that Mayo protocol that we talked about. And for those that do fit the criteria and did undergo the Mayo treatment, survival at 1 year is 88%, and 5 years, 82%, which is quite good. And recurrence-free survival post-transplant is about 65% at 5 years. Changing topics a little bit, even though we do liver transplant and we think it's an incredible option, clearly we know that there is a growing gap between supply and demand. And even though we're transplanting more people than ever, we're also listing people more than ever, and so that gap has increased. Which brings me to thinking about living-related or living non-related liver transplant. I am going to just say a little shout out to my institution where we did perform the first live donor liver transplant in 1998. And the reason live donor liver transplant is a good option for everyone to think about is, one, we know the MELD score is not perfect. It frees you from the allocation system and being on the wait list. It allows you to get transplanted before you're really, really sick in the ICU. It's somewhat an elective surgery, so you can time when it's going to be done. Donor organs tend to be of higher quality than organs that are from someone who have died. And at least at our institution, we're now doing lap-assisted and lap hepatectomy to the donor. So the recovery time for the donor is much, much less now that we started doing this. And as we'll talk about, there's excellent survival. And you can see here in this figure that since we did introduce live donor liver transplant, there is a great survival benefit compared to pre-live donor era. So this is a study that recently came out that looked at patients that had PSC and how did they do with live donor compared to deceased donor. And you can see from both of these graphs, which show both patients' survival and graft survival, that survival is comparable. In fact, patients that were transplanted with live donors had actually better outcomes with live donor than with deceased donor. And importantly, PSC recurrent rates does not differ between live donor or deceased donor. So I'm just going to end with a couple of things that are new and exciting. We recently published a study where we demonstrated that the benefits of live donor clearly outweigh the risks of live donor for patients with MELDs of 11 or above. And so maybe we should not be thinking of this MELD 15 rule as when we should start thinking about transplant, but it can be lowered to 11. And regardless at what MELD score someone is transplanted at, they do gain an average of 15 life years. Another thing that we have at University of Colorado and some other programs in the United States are doing this also is non-directed or anonymous live donors. And so these are people that just will call up our program and say, I want to donate. And if you look at our program so far, we've had 66 non-directed liver donors. Interestingly, 15 of them had previously donated a kidney. And of those 66, 33 have successfully donated. And we've completely eliminated our pediatric waitlist by the use of these anonymous donors. We also are able to use normothermic machine perfusion, which is a technique that takes a marginal organ and makes them what we think to be optimal. So after a donor has died, we basically hook up the liver and supply them with oxygen and nutrients. So this we think is going to impact our survival. And we're also doing now paired exchange in chains like they do in kidneys. So if I am a blood type A, but my donor is a blood type B, in the past we'd say no go, but now that donor B can donate to someone else and I can get someone else's donor. So we think that this is pretty exciting too. So in summary, I think it's important to do your research and ask lots of questions. It's a multidisciplinary approach. Liver transplant for PSC has excellent outcomes. Recurrent PSC, though, does occur in about 20%. And it is an option for select patients who have cholangiocarcinoma. I think live donation is really an excellent option that you should talk to your hepatologist about. And then the last I can't stress enough is that field of transplant is rapidly advancing. Thank you.
Thank you, Lisa. Now, Ricky, I think you, me, and Josh are supposed to. Josh,
He's right.
come on up.
All right, I guess the three of us are going to sort of
Do a round.
close up. Yeah,
Close out.
we'll each make our comments about the whole experience. Okay. So the time has flown by really too quickly for this weekend, but it's been a fantastic experience for our community to gather in person again to hear the weaving of perspectives of all these top international PSC experts. As patients and caregivers who live with this disease on a daily basis, we feel an incredible urgency for the development of better therapies and eventual cure. The unmet needs for our unpredictable disease are overwhelming. We are extremely grateful for the clinicians and researchers, both academic and industry, and the regulators who are here today and who share our passion. Thanks to the expert leadership of our two co-hosts, Dr. Korzenik and Dr. Bowlus, along with our CSO, Dr. Rossi, and insights from our wonderful international speakers, together we have accomplished what we set out to do. Present an overview of the current state of PSC knowledge and global PSC research, champion the patient voice as we continue to break down silos and other barriers slowing down research, spark multi-stakeholder collaboration, and facilitate patient-driven projects and patient-focused drug development. This is a really exciting first step in the in-person launch of our ICR network. Beginning in 2005, when PSC Partners was established, we recognized that in order to be successful, we would need to build key relationships that would lead to collaborations with multi-stakeholders working in the PSC space. The first in-person meeting of the ICR network has inaugurated an open dialogue among patients, caregivers, researchers, industry, and regulators to reach our shared goal of accelerating PSC research. With dedicated commitment from all stakeholders to work together, we can overcome the current gaps in research and drug development. At PSC Partners, we are eager to pursue this initiative. So these past 2 days, you've witnessed the results of the passion and tenacity of our patient caregiver community. At PSC Partners, we're actively driving research forward, and we need continued input from researchers, regulators, clinicians, and industry partners to help us develop a prioritized research agenda that captures what this group here recommends to be an effective road to a cure. Over and over through the years, we've witnessed the commitment of scientists who have formed powerful networks in their search for a cure. Our vision is by including patients as active participants whose passion will tirelessly nurture the program we build together, we can solidify our search for a cure. We believe that the hours we have spent together striving to formulate a research agenda will bring the same passionate determination to achieve the program we create. So we ask you, patients, caregivers, researchers, regulators, clinicians, and industry to stay with us and to continue contributing your valuable input. Together, we are creating a world where a PSC diagnosis comes with a cure. Thank you all.
Go for it.
Thank you very much, and just want to thank you all for being here. When we were planning this conference, I was worried that we were trying to do too much. The time was too short, there was a sense of urgency, but we accomplished what we wanted to do and more. We had great scientific discussions before. We heard numerous voices from whether it's pediatric, gastroenterologists, hepatologists, whether it's from the FDA, whether it's from bench researchers, clinical researchers, translational researchers. So that was extremely productive and effective. And then having all of you join and really hearing the voices of patients and understanding where everyone is coming from, sharing advances, sharing an understanding of where we stand with the field right now. So that's been extremely helpful, extremely productive, and very exciting. I'm very grateful also to the Resnick family who are funding our research, and we really want to shepherd those resources to be able to help the broader community and really create a collaborative environment and help grow that. When we think back, perhaps 20, 25 years ago, if we were sitting in a clinic and we might see two patients, one with, say, hepatitis C, and we would sit there, and one with PSC, we would say to both of them, well, there's not much we can do, really. We'll see what we can do. Maybe there'll be some research. Maybe there'll be some advances. We don't see hepatitis C as much, really, anymore. We have cures. That's now vanished. We're still with the PSC patients saying, well, we're trying to do research. But I mention this because we see how transformative things can be, and how quickly we can really take a disease that we don't understand and make it so that it is relatively simple to manage and to cure. So even though I agree we're hesitant to use the term cure because we don't want to create false expectations, and some people say, well, if we can just shift the curve a little bit, that's what we need to do as an initial step. And that's what Rich Green was really suggesting, is something that's effective that can help manage things. But I think that PSC is so confusing and is so, I'm sure when you hear some of the research out there, it's kind of bewildering because fundamentally we don't understand it. Once we understand it, and I really do believe we will in the near term, it's not going to be so confusing. And a lot of the different parts, sort of like the blind people feeling the elephant, it all seems different to everybody. We'll see the whole picture and things will make sense. So I encourage everyone to be involved. Like the conference, the time is short, but the need is great and the time is urgent. So thank you all, and we will get this done. Much appreciated.
So I can keep talking for another 20 minutes, or we can end early. Give me 2 minutes, okay. I just also want to thank everyone that's participated in this. And shout out to Josh and Ricky and Steve Rossi, who really brought this all together, both on the ICRN part, which was a new initiative, as well as finally getting back together in a patient conference. These are just so energizing for, I think, us on the other side. And as much as you get out of this as patients, I think we get out of it being here with you. We've been doing this for a long time, and watching where PSC Partners has gone from when we first got involved, and I think it was early on that I got involved with you all, and we were doing research, some research grants, and having the patient meetings, to where we are now with WIND and the symptom assessment project and the ICRN. It's sort of like it's been this slow, gradual increase in progress, but we're kind of, I think, at an inflection point where we're really seeing this incredible movement. And I mean, that's what makes us, I think, optimistic when we talk about having a treatment in 5 years that may be effective. Because I think we've got the science going on, we've got the research infrastructure, we've got a plan in place, it's continuing to evolve. And I think the future looks really bright for all of us. I know that a lot of people here are new to coming to these conferences, and it may seem like it's a lot of research, and maybe that's not what you're looking for. You're looking for how am I going to do with my PSC? But as your journey goes on, I think you'll appreciate that the research that's being done is really what's going to then bring hope and treatments to you all over the coming years. So again, thank you all, and really been a fantastic conference for me. I've really enjoyed it. And I think we'll close the session at that time, or do we want to have questions?
Do you want to, I guess probably do a Q&A. We have time, don't we? Yeah? Do you want to do Q&A?
Announce about the change.
Dinner will not be outside. Is
Yeah,
that the
I told
change?
them at lunch. Yeah. I don't, Joanne, are you going to do Q&A? Are you collecting notes or no? Are there questions? Raise your hand if
Raise
you have
your hand if you have a question.
I know everybody's exhausted. We may be too tired for Q&A.
We're done? Well, with that, excellent.